welcome, and thank you everyone for joining. We're looking forward to a great discussion this afternoon. I am Michael Yusheko. I am the Executive Vice President and General Manager at Tempus. I've been with Tempus for about 6 years. I help to lead and support our core genomics portfolio with our provider, clinical partners, as well as our life science teams. Thank you again for joining. The reason and the conversation that we'll have over the course of the next 45 minutes to an hour really stems from more recent announcements that we've had around our expanded liquid biopsy panel, XF Plus. And in light of that, we wanted to host this conversation really around the broader evolution of liquid biopsy in both clinical practice and development. And I have the pleasure of being joined by two of our esteemed colleagues, Nikki Bouvier and Ezra Cohen. Nikki, if you want to quickly introduce yourself.
Yeah. Hi, Nikki Bouvier. I'm SVP of Life Science and Pathology, and my background is as a surgical and molecular pathologist. I've been with Tempus for about 7 years, working in clinical and then in R&D.
And Ezra?
Hey, thanks, Mike, and thanks to everybody for joining. I'm Ezra Cohen. I am Chief Medical Officer of Oncology here at Tempus. I've been at Tempus for almost 3 months now, so really getting excited about what the possibilities are and happy to be part of this webinar. Thanks.
Great, thanks, Nikki and Ezra. All right, so let's just jump in and get into the meat of it, and we'll certainly hope to retain some time here at the end to answer some questions as well. But maybe just to open, how has the use of liquid biopsy in both research and the clinical setting kind of evolved? And maybe Nikki, we'll start with you first.
Right. Well, the main evolution has been, first of all, its existence at all relative to tissue testing, which really has happened in the last 5 years or so. And in addition to that, the size of the panel getting much bigger, that's due to technical improvements and cost of sequencing coming down. So now we can offer a 523 gene panel, which is a large subset of our 648 gene solid tumor panel. And we can do things like cover all of the homologous recombination repair genes. We also have enough genomic space on that panel to calculate tumor mutational burden. A lot has been written in the literature that you need probably about 300 or more genes to do that robustly. So being able to offer these bigger panels in the liquid space is relatively new. It offers the possibility of being able to monitor patients longitudinally when it's infeasible to get repeat biopsies. And it also has, it's not just that it's difficult to get a biopsy, it's also that liquid will sample from all metastatic deposits that are shedding tumor and not just from the one particular one that you put a needle in. And that gives it a good view of the emergence of resistance mutations and response to therapy that you can see through the overall tumor burden as measured by variant allele fraction or circulating tumor fraction estimate. Because of these characteristics, you can sometimes find mutations in liquid that you don't see in tissue. That can go the other way as well. So really the combination of tissue testing at diagnosis plus longitudinal monitoring with liquid biopsy gives you a very comprehensive view of the mutational landscape and the response to therapy over time.
Great, thank you. Ezra, anything to add there?
I think Nikki makes some wonderful points, and I totally agree with everything she said. It's interesting when these assays first came into practice. I think many of us had to understand how to use them. Maybe were a little bit reluctant to believe what they could deliver. And then over time, we've learned to integrate them into our practices and for that matter, into our clinical trials, which I know we'll talk about a little bit later. And I completely agree with the point that Nikki made about the complementary way to use both the tissue and the liquid biopsy, in that often we do find a different set of mutations on those two different types of assays. And if you think about it, the tumor biopsy is sampling a small piece of a heterogeneous tumor in a heterogeneous process. Meanwhile, the liquid biopsy is, in a sense, sampling what is being shed from the entire tumor mass into the blood. And so I completely agree. I really do see these as complementary tests that give us more information about that patient in front of us and allow us to better individualize the therapy and be more precise. And, I always say to patients, and for that matter, drug developers, researchers, the more information we have, the better off we will be in terms of therapies, planning, execution, et cetera. And the nice thing about the liquid biopsy is now we're at a point where if a mutation is found on the liquid biopsy and not on the tumor, we believe it and we can act on it, and we often do. And in fact, I can think of a lot of patients where I've done exactly that with good effect. Of course, the liquid biopsy is a little bit faster to develop, so we can get the information a little bit quicker, maybe a week or so, maybe a little bit more than that, than the tumor. And for the most part, in terms of getting a result, sometimes we do know that for the tumor biopsy, there's insufficient tissue. Something may go, in terms of storage or processing. Something may not be ideal, and sometimes with quite honestly the tumor sequencing, we don't get a result. Meanwhile, that is less common, I would say, with the liquid biopsy. And then finally, I think the impact in terms of patient care, it's as I mentioned, it's procedurally faster. It's certainly less invasive, easy to get a blood test on the great majority of patients, and it's more accessible. So now we do have mobile phlebotomy. These tests can be done at the patient's home. They don't have to go to the office. They don't have to travel, put themselves potentially in a compromising position, especially when many of these patients may be immunosuppressed due to the therapy that they're getting. So really, again, I see them as complementary. I think there are definitely advantages to liquid biopsy. And I think at this point, in terms of practice, the great majority of oncologists have really accepted this as part of practice in the oncology patient.
Thanks, Ezra. So Nikki, maybe looking over to you just on the subject of the impact of patient care, just a bit curious, how do you think that this evolution of liquid biopsy that you both have kind of spoken about has impacted and is now kind of as a tool in clinical trials specifically?
Right. Well, for clinical trials, I would say there are two aspects to it. One is operational and the second would be scientific. So operationally, it's not always possible to write in a repeat biopsy in a trial. And even if you could, it's expensive, it's difficult. And as Ezra mentioned, there are a lot of reasons why it might be undesirable for the patient if someone is neutropenic or immunosuppressed, or if the tumor is just at a difficult site. Even, lung is a very common use case, lung cancer, and smokers have COPD. They can get pneumothoraces from biopsies. So there are a lot of operational reasons why it's easier, and therefore you would have greater compliance if you go for liquid instead of trying to do serial tissue biopsies. And it's feasible to get them a lot more often. And that being able to get them often, or at whatever cadence you would like, gives you the opportunity to monitor a drug response, both with emerging resistance mutations and also with the overall tumor burden. And that way, the group running the trial can really get up-to-date information about how it's going without having to wait a long time to assess it. So I think that's valuable in terms of making clinical trials faster, more nimble.
Ezra, maybe switching to the clinical perspective from your experience running trials, is there anything else you would point to as having evolved or improved in that space due to the emergence of liquid biopsy?
I think Nikki touched on some of the aspects of the tests that allow us to begin to integrate these into clinical trials and really use the information meaningfully. So first, there's the aspect of identifying patients based on the liquid biopsy. We used to rely completely on tissue. And I think as the tests evolved, as they got better, as researchers really got comfortable with using them, now many of the clinical trials that rely on a specific biomarker, and that biomarker could be a genomic level alteration. In some cases, it may even be a fusion. In some cases, it may be, Nikki mentioned TMB and the ability to assess TMB through the liquid biopsy. We can now use the liquid biopsy to identify patients and determine eligibility. So that's one big factor. We don't have to do that biopsy again. If the tissue isn't available, we don't have to worry about it. And as we mentioned before, it's quicker. The other is it can help begin to stratify patients. We can, and there are clinical trials now that are looking at the absolute level of circulating tumor DNA in some for some specific alterations, and also the change over time. For instance, this could be used in the neoadjuvant setting to stratify patients for subsequent therapy. We could also use the assay after the completion of therapy, perhaps in the curative intent setting, to look for patients who may be at higher risk for the endpoint or the entity that's being studied. And then lastly, as Nikki, I think, summed up very well, it can be used to monitor patients, especially for patients who are going to develop early resistance, and certainly for specific targeted therapies, this is already being used. These resistance markers or resistance alterations often develop months ahead of clinical progressive disease, especially disease seen on radiographs. And once that develops, one could think about an early intervention to extend that disease-free survival interval. And of course, there are clinical trials that are doing that right now. The nice thing for drug developers is that regulators are listening to this. They are beginning to understand how these assays can be used. And although I don't think we're at the point where for some of these case examples that I gave, approval is ready to be made on that, but we will get there. And in fact, for the eligibility aspect of it, regulators have accepted that, have accepted a liquid biopsy for many of these biomarkers or many of these alterations for eligibility. So we're going to get to a point where these liquid biopsies are heavily integrated into clinical trials and eventually the approval of newer agents.
And as we're staying with you for a minute here, what are the potential benefits of ctDNA-based therapy response assessment in addition to traditional methods like you were mentioning, such as imaging or tumor markers?
Yeah, I think we can take a page here in solid tumors from what our hematologic malignancy colleagues have been doing for arguably a decade, and that is we get an early look at the effectiveness of the intervention with these types of assays. In a sense, we can view it as a measure of molecular disease that is going to be much more sensitive for the state of disease and a much earlier readout than with traditional radiographs and certainly with clinical examination. So those really are the two main advantages I see, that we can assess at the molecular level what's going on with the disease. Remember, these are quantitative assays as well as identification of the alteration, and we can track that alteration and new ones that develop that portend eventual resistance, as I was mentioning before. This is often used in patients who are being treated with EGFR inhibitors for non-small cell lung cancer. And that switch to therapy earlier does seem to have a benefit for patients. And not only that, we can begin to think about it for discovery, for certainly novel agents and potentially novel mechanisms of resistance. And we can begin to think about detecting these on circulating tumor DNA in real time rather than having to do a biopsy. And not only that, when we think about immunotherapy, well, that really opens up a whole new world or new way to use circulating tumor DNA, because what we're challenged with in immunotherapy is that assessment of response for really two main reasons. First of all, immunotherapies can take a long time to manifest a change in tumor size on radiographs. Sometimes we see it as early as 8 weeks, but sometimes, and in many patients, we don't see it for months. So an early readout of what's going on with that therapy and if that patient is benefiting would be tremendously beneficial. And secondly, we can also assess whether that patient in front of us is having a reduction in the circulating tumor DNA, because if they're not, it really does appear that they are unlikely to benefit, and we don't have to expose them to that toxicity for a longer period of time. So in the world of immunotherapy, I think circulating tumor DNA is going to prove incredibly beneficial.
So it looks like we have some good time here left for questions. So I'll just maybe open it up to the Q&A section, and we have a few questions here that have already come in. So I'll open up with the first one. Apart from oral selective estrogen receptor degrader development, what are other applications, practically speaking, of liquid biopsy?
Yeah, maybe I can take a stab at that, Mike. So I think for things like for diseases like prostate cancer, we can begin to, especially in the castrate-resistant metastatic patient, we can begin to think about using the liquid biopsy to identify patients who may be good candidates for, let's say, PARP inhibitors in combination with androgen receptor blockade. Certainly for patients who are androgen receptor therapy refractory, we can identify patients with homologous recombination repair or some sort of DNA repair defect that may be good candidates for those classes of DNA targeted agents like PARP inhibitors. And of course, as you know, there are many more agents in development hitting other targets in these pathways, ATM, ATR, et cetera. So prostate cancer, castrate-resistant prostate cancer, a disease that we think of as relatively refractory, we can identify a group of patients that may be quite responsive to certain agents. And then going beyond that, I think most cancer types can benefit from liquid testing, especially for trials with targeted therapies. And it really is, as I mentioned, it's a great tool to detect these alterations that may be missed by tissue. It's a great way to monitor these alterations, and it's a great way to look for new alterations that are going to come up that may portend resistance way before the patient actually progresses. It really opens up so many possibilities. I hope that answered the question.
I'd actually like to jump in as well, building off what Ezra said, and in particular with prostate cancer, thinking about actually getting the information to the doctors and patients. One of the issues we face with prostate tissue, it's well known in the field and never been understood, but NGS sequencing fails at a pretty high rate in prostate tissue. There's probably some sort of PCR or other inhibitor there, but people have tried millions of different ways to get it to work better, and it just has a high fail rate. So that's something where you can reflex and get an answer from the liquid biopsy. And because this panel has 523 genes, you're not losing that much information relative to a large panel tissue test.
Yeah, it's a great point.
Thanks, Wes. So it looks like we have a question from a partner that we've worked with in the past. We've used Tempus's XF plus liquid biopsy before. How is it different than XF?
I can take that one. It's a pretty quick answer. Basically, XF is 105 genes, so it's the really common and actionable driver genes. And it does not, as mentioned before, you really need about 350 genes to do tumor mutational burden properly. If there are too many driver genes, it just massively overestimates the TMB. So it's not possible to do that with the smaller panel. So the bigger panel, you have 523 genes, plenty of genomic space to do TMB, and you get a lot more genes to play with. So not just the really big clinical drivers, if you're doing any kind of research or pathway analysis, looking for some more obscure resistance mutations, it gives you that whole genomic space to play with.
So we'll go to the next question here. So great question. Do we really need NGS for companion diagnostic applications, especially when all you might care about is an EGFR point mutation?
I can take a first stab at that. I would say yes and no. So first, I assume the questioner is really focusing on lung cancer, because that's where we often see the actionable EGFR point mutations or variants. So first of all, for most patients that come into the clinic, we don't know if there's an EGFR mutation. The clinical characteristics may favor that, but as we know now, those are the same clinical characteristics that favor other driver mutations or driver fusions such as AKT, RAF, et cetera, et cetera. So one advantage to a broader panel is that you may find other alterations that are actionable that you have no way of otherwise knowing. Now, let's say in a world where, for whatever reason, you knew this patient had an EGFR driver mutation, I would still argue that there's room or interest in getting a broader panel for a number of reasons. Although it's unusual, but it's not impossible to have secondary alterations or co-occurring alterations that may inform your therapeutic decisions. I agree that's not a common scenario, but it's certainly a possibility. And I believe that one would want to know that or exclude that. And then secondly, of course, as we talked about in the discussion, especially for EGFR point mutations, I think one would want to see if secondary resistance alterations are developing in not only in EGFR, but in other pathways, because that would, and we will have data on this, we have some data already, that should signal at least a discussion about changing therapy, if not actually changing therapy. So I think there is a lot of value to getting a broader panel rather than just looking at one gene. I don't know if Nikki or Micah, if you want to add anything to that.
Right. I mean, I would say I totally agree with that. And just to be a little more specific, even there are some emerging biomarkers in lung, like HER2 we look at now in lung. The panel does HER2 amplification, can detect that, and STK11. There's rarely, but it does happen that there are some other mutations in the MAP kinase cascade, even if you have an EGFR mutation. So you don't tend to see like a codon 1213 KRAS mutation plus an EGFR, but you will see sometimes things like an NRAS codon 61 mutation, one of the, I would call it, not the technical term, but like a milder MAP kinase mutation. So if you really want a comprehensive view of what's going on, I would say you do want KRAS, BRAF, NRAS, MEK, ERK, PIK3CA, basically the whole gamut of that pathway. And then you also, of course, want to see P53, what's going on in that pathway, and if mutations are accumulating there. So, day to day managing one patient who has a T790M, maybe not, but in aggregate, you don't know what you're going to find. And especially if anyone has a research interest or a clinical trial interest, I think you would want that more comprehensive view.
Next question here. How is Tempus addressing the issues around clonal hematopoiesis and contamination of signal with biopsy, in particular for genes like ATM and TP53 mutations?
I can take that. So it's a good question and it's a hard question. I think the absolute ideal way to do that would be to sequence the Buffy coat and subtract it out, sort of analogous to the tumor normal subtraction that we do out of solid tissue. However, that's not feasible for really, frankly, cost reasons. We sequence these panels super deeply and they're difficult to run. So instead, we've looked at bioinformatic ways to handle this. And one of the things, we have a database, of course, where we're tracking these, and it's well known in the literature. So the ones that look heme-ish, like JAK2 or DNMT3A, it's pretty easy to tell that those are CHIP. The issue, as the person who posed the question rightly said, are genes like KRAS and P53 that are common driver mutations in the tumors you're looking for. So there are a couple ways. I mentioned before that one of the things we really like to do with this panel is to get an upfront paired solid tumor and liquid biopsy, and then follow longitudinally with liquid biopsy. So what that gives you is a baseline in the tumor. If you see something like a KRAS or P53 mutation up front in solid tumor testing, then you know that's a real tumor driver. If you don't see it there and it pops up later in liquid biopsy, then that's very likely to be CHIP, because generally tumors don't evolve a new driver KRAS mutation halfway through treatment. So there are ways, in other words, there are databases that we can use. There is the upfront solid tissue testing, if we have that to compare. And then the other thing is a careful analysis of the variant allele fractions. Often the CHIP genes will be at a higher variant allele fraction than the tumor genes, or maybe the opposite, I guess, depending on the tumor burden, but you'll see them sort of at a different level, so you can take an educated guess there. But it's a good question, and I don't think it's 100% solved.
Great, thank you, Nikki. Next question here. How do we go about cases where mutations are identified in the tumor sample, but not in the liquid biopsy, or vice versa?
I can maybe take a stab at that first. So this is really not unusual at all. In fact, we commonly see an alteration showing up in one and not the other. What we now understand is they are both real. They happen for mostly known reasons. Sometimes we don't completely understand why, but my approach is, and I think in general, to believe the data. So if, for instance, and I'll give you a real world example that of a patient I'm treating right now, their tumor tissue NGS didn't show any driver or let's say actionable alterations, while their liquid biopsy showed a BRAF fusion. And in fact, we were able to get that patient on a clinical trial, specifically with a BRAF inhibitor that seems to be more effective in class 2 alterations, and that patient is responding wonderfully. That is, was not seen on tumor tissue, but we believed the liquid biopsy, the clinical trial allowed inclusion of that patient based on liquid biopsy, and now that patient is benefiting. And certainly, there are many examples of the converse also being true. So getting back to what we were saying at the beginning of this webinar is that really these are complementary tests, and the way I would approach an alteration that is seen in one and not the other is I would believe it and I would act on it. And I can tell you from a lot of anecdotal experience that is valid. I don't know if Nikki or Mike, you have anything to add to that?
No, I would totally agree with that. And I, from my perspective as a pathologist, I think there are different pre-analytical factors that can influence that. We talked a little bit about how it's tissue testing in prostate can be difficult. There's tumor heterogeneity. There are a lot of reasons why something might be seen in tissue and not liquid. If the tumor doesn't shed well, for instance. And then on the flip side, you're sampling a small amount of tumor. The biopsy may have a very low tumor percentage if it's heavily infiltrated with lymphocytes or other immune cells, but you may be able to pick up that mutation that's shed into the blood. So given that there are sort of pluses and minuses and pre-analytical variants to both assays, I really like concatenating the two data sets to see what you have. I think it gives you another shot at it.
Moving quickly, it looks like this question is a little bit more on the clinical trial side. So to be informative, how often should the sampling be done during a clinical trial?
Don't know. but so there's no definitive answer. There's no gold standard. And the reality is that we are still trying to understand fully the dynamics of change in circulating tumor DNA. I can tell you that it varies by the tumor burden, it varies by the disease in question, and of course, factors related to the individual patient. So there is no known perfect answer to that question. Having said that, I think many of us look at about a 4-week interval. In some cases, shorter than that, and certainly I can say with an intervention, we see changes even within a few days, depending on the intervention. But what often happens is that we see an increase in circulating tumor DNA early on, reflecting some effect of the therapy that either continues to go up when the therapy is ineffective, but hopefully starts to drop and drops below baseline when the therapy is effective. That sort of drop we will see in the great majority of patients at around 4 weeks, and that's become the acceptable time period, that 3 to 4 week interval from baseline, for a couple of reasons. One that I just mentioned. The second is practicality. Cycle lengths are usually 3 to 4 weeks. That's when the patient's coming in again. You don't have to ask the trial participant to come in for a special visit. And then beyond that, that really, I think, depends on the hypothesis being tested in the trial, the reason to do the assay, and of course the disease and intervention in question. I've certainly seen trials look at circulating tumor DNA fairly regularly, even at monthly or bi-monthly intervals. And I've certainly seen trials where it's that 4-week or 6-week measurement, and then, for instance, at progression. So I think subsequent to that initial, it really depends on the specifics of that study.
So next question, do you have a framework for thinking about which diseases are better candidates for liquid biopsy? For example, small cell lung cancer would seem to be a great candidate because of high nucleic acid shedding. However, liquid biopsy is still evolving in this disease area.
Nikki, do you want to take that, or you want to go first?
Sure.
I've got some thoughts as well.
So the question was small cell, so high grade neuroendocrine, correct? Yeah.
Specifically calling out small cell, yeah.
Small cell, right. So I think the issue with small cell is that the most common mutations we see in there are P53 and RB1. Those obviously you can pick up. I guess the question is, what's the actionability of P53 and RB1? You can certainly monitor disease burden. So if they're getting radiation or other treatment, you can monitor how they are doing on that treatment by those variant allele fractions going up and down, or the circulating tumor fraction estimate, which is even a more robust way to do it. I think the question might be getting at, what about a disease where there isn't a highly targeted therapy? Is it worth doing that? And I think, the answer is that it depends. As Ezra mentioned, if you want to monitor somebody every 4 weeks or every 6 weeks, I don't think reasonable to get a biopsy that often. So liquid gives you the way to do that. And there are driver mutations in those diseases that are trackable, if not actionable.
Yeah, so I would agree. I think it comes down to the same principle that we talked about at the beginning, is you don't know what you don't know. And so even in a disease where it may not be common to have actionable alterations, we still see patients that have them for different reasons. They may be DNA repair defects. They may be high TMB, which of course, is now associated with the therapy. They may be something else. And so you don't know if you don't check. So that's the first thing I would say. The other is, I think for the spectrum of cancers that one could see, there's, in my mind, really, there's always a reason to justify getting the liquid biopsy. In a disease where we expect to find driver alterations, that I think is fairly obvious and intuitive. In diseases like sarcoma, where we don't expect to see them, I think there's still a value to getting circulating tumor DNA for some of the reasons that we said. First of all, the possibility that there may be a driver that we simply don't know about. The ability to monitor therapy and the effectiveness of it, the ability to get a early readout of either progression or response, and even the ability to detect emerging alterations. So I think for the spectrum of cancers, there's widespread utility for these types of assays, and in fact, I think that's exactly what we're going to see going forward, is that the liquid biopsy is really going to become almost a common practice, even I dare say a standard practice in almost every patient, if not every patient with cancer.
Thank you. Next question. So this is going a little bit back to some of the discussion that we had earlier, just around VAF and CTFE. So question is there any difference between VAF versus circulating tumor fraction estimate? I read a white paper from Tempus that reports CTFE.
I will definitely let you take that one, Nikki.
Yes. So the difference is median variant allele fraction is what it sounds like. It's a calculation of the median of the mutations that you see, including VUSs. It's vulnerable to a couple sources of error. One of them is CHIP, and the other one is germline. It's pretty easy to see which mutations are germline because they're basically hanging out around 50%. But if you have CHIP mutations coming in there, they can throw off that calculation. And also, if the patient doesn't have any mutations at the moment, you won't have anything to calculate. Circulating tumor fraction estimate, I like a lot better. The Tempus name for that is Otter. We managed to contrive it so that it would have a cute animal name, but anyway, it's circulating tumor fraction estimate. And it uses off-target reads throughout the genome to calculate to see the difference between the profile of the tumor and the normal DNA. So it's more robust to artifacts and different issues with calling specific mutations. So it gives you a picture of the whole genome, and the output of that will be something like there's 2% circulating tumor.
Hey, Mike, before we get to
Appreciate
the next question, I see and I'm getting messaged about the patient that I described earlier with the BRAF fusion. If they were metastatic, indeed they were metastatic. Just to clarify that, so they had a fair bit of disease at several sites.
So next one, just looking forward to the future. Do you believe that liquid will take over IHC or ISH biomarkers?
No.
Yeah, I think they're go ahead, Nikki. Yeah.
Yeah, no, I think your whole career as a pathologist, people are like, oh, this is going to happen, it'll replace you. It's like, no, I have more work than ever. It's always additive. Things like IHC and in situ hybridization, those are looking at things like, or especially chromogenic in situ, those are looking at proteins. So you're not going to replace a protein assay with a DNA assay or a DNA assay with a protein assay. They're looking at different things, they're complementary, and they're both great sources of information.
Yeah, I would agree. To be a bit provocative, it may replace our use of radiographs, CT scans, et cetera, if we have essentially molecular assessment of disease status and response to therapy. I could imagine a day where we don't get as many CT scans to monitor patients, especially ones where we're interested exclusively in their response to therapy, but we're not there yet. That may be the future.
Very good. Very good. So people could sleep well at night on the pathology side. So next question, will the XF panel suffice to identify HRR mutations in MPC?
I assume MPC is metastatic prostate cancer, or is it pancreatic, or do we know from the questioner?
We don't, but
because the answer is yes. I guess irrespective of the disease. I don't know, Nikki, if you want to add to that. So yes, the XF and XF. Well, actually, XF plus is probably that's a better use for XF plus because it covers the broader array of DNA repair genes. So I would say XF plus, yes, in both metastatic prostate and metastatic pancreatic, or whatever the P stands for.
Yes, totally agree with that. And again, BRCA and the RAD51C and so forth, the common ones are in XF, but the full array of HRR genes are in XF plus.
So yeah, I would say if somebody's really interested in that process, I would get XF plus.
Very good, makes sense. So next question's coming in, just trying to draw some parallels, looking at liquid and tissue. So what are the main differences between liquid and tissue somatic DNA sequencing at present in terms of read depth, accuracy, chip? And then the second part of the question is, how do you measure TMB in liquid biopsy?
I can take some of the technical aspects of that. So, sorry, was the first part of the question, Mike?
Just looking for the differences between liquid and tissue somatic DNA testing in regards to read depth, accuracy, and chip.
Right. Okay. Yeah. So the read depth that you need to sequence to is determined by what you want your sensitivity to be. So the cutoff for the lower limit of detection for tissue is 5% variant allele fraction. That's what we're validated to. We can go down to about 2 or 3% for certain hotspot and resistance mutations. So to do that, sequencing about 500x deep gets you that comfortably. On the flip side, with liquid, where you're trying to go down to 0.25 variant allele fraction with maybe 0.1 in hotspots, you have to sequence 2,000 or so unique reads to get to that. And because of some of the technical reasons, when you're sequencing that deeply, you get higher duplication rates. So the raw reads that we get are really like 20,000 or so deep. So to get to that 2,000 deduped reads. So it's really just a pretty straightforward calculation between what variant allele fraction you want to get to and how deep you sequence. I would say the solid tumor testing, especially if you have the normal match, is very accurate because you get the germline subtraction. You don't see any chip with it. Actually, that's not true. Occasionally in really bloody biopsies, you can see chip, but we get rid of that. Anyway, it's a clean data set of somatic mutations in that piece of tissue. That's ignoring tumor heterogeneity, of course, it's in that tissue. On the liquid biopsy side, as mentioned, we do see chip, we don't do germline subtraction. We do a little bit of germline subtraction, but it's based on population databases. So in certain ethnic populations, that's not as effective as others. And so you do see those things in liquid biopsy. You can look at the normal sequencing from the tissue and compare it to the liquid to clean up that data set. So again, the combination of the two data sets is going to get you the best information. But that extreme sensitivity, that very low variant allele fraction, that's just due to the super deep sequencing.
Last question, then I'll move to, which I think is a good one because it just looks forward to the future. So just bear with me for a second as I work through the question. So I feel like there are so many other extensions of ctDNA and cfDNA. As a genetic counselor, we have detected metastatic cancers on NIPT prenatal tests and on germline panels when we see low AF mutation signals. So ctDNA along the way of a cancer patient journey from ctDNA for multi-cancer early detection to monitoring and survivorship. Can you comment on future directions of liquid ctDNA?
I think the questioner, provided the answer. It's a great question, by the way, and a great way to think about where this field is going. I think. And we talked a little bit about this. I think it will be again a way part of the standard practice in assessing a patient at that first visit. I think it will be standard way to monitor tumor burden and treatment response. I think it will give us an early readout much earlier than radiographs, and then for patients treated in the curative setting or in the minimal or in the no evidence of disease setting, I think again these assays will give us an opportunity to improve surveillance and detect recurrence sooner than conventional methodologies. The questioner, points out potentially other instances where these fragments could be found, which certainly may play into management. Obviously not routine cases in oncology, but as this technology really begins to become wider and wider utilized, I think we're going to see cases like that. So it's a really interesting scenario that the questioner points out. But I think, yeah, this is just the beginning.
Vicki, anything to add on your end before we wrap?
Yeah, I mean, I think it's interesting. It's more data is always great, but also has burdens that come with it, right? So on the flip side of that, we see a lot of incidental germline findings in diseases in our, say, tumor normal match that are related to things like maybe cardiac risk and so forth that you weren't necessarily looking for in a cancer panel. So I think you just have to have really alert clinicians who can take all this data on board and follow it up. But yeah, to me, anything that uncovers a disease earlier, gets you something that you can action on, that's fantastic.
We really, really appreciate all of the questions, the dynamic conversation. Nikki and Ezra, really appreciate your time, and for our audience on the line here, thank you for spending the last hour with us, and in particular, all of the great questions that came in. We got through as many as we possibly could, tapping all of our time. So appreciate you staying with us, and hopefully you enjoyed the conversation as much as we did on our end. So from all of us on Tempus, thank you for joining us. And for those of you who are interested in joining our next webinar, there's some information here up on the screen. Please feel free to scan that QR code that's on the screen so you can get some more information and register for it, Tuesday, August 22nd. And we'll look forward to spending more time with you. All the best out there, and look forward to hopefully having a chance to speak with many of you here in the near future. Thanks so much.